Okay. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. Assalamu alaikum wa rahmatullahi wa barakatuh. You don't have to share the screen right now. Just wait until I tell you to start sharing, okay? Inshallah. Today, the date is October 29th, 2025. The time is 10:46 a.m. Eastern Daylight Time. We have with us Muhammad Irfan Alam. He is from India, Bihar. He is going for his diploma in artificial intelligence operation, EduQual level 6, RQF level 6. The topic he has is orchestrating synthetic biology automation platform with BioCAD, OpenTrons, and laboratory robotics for biotechnology research, right? The project has multiple facets. There is synthetic biology design automation, laboratory robotics integration, bioinformatics pipeline management, laboratory information management system, LIMS, metabolic engineering and optimization, and regulatory compliance and quality management. You have to design a solution using open source tools, cloud native tools for robotics, bioinformatics, LIMS, modeling, data management with Apache, Airflow, MinIO, Postgres, SQL, so on and so forth. Along with mentioning some of these standards, which are mostly the US standards, the FDA 21, the CFR Part 11, so on and so forth. In this presentation, you'll be given minimum 15, maximum to 20, 25 minutes, followed by an interview of 30 to 40 minutes. The whole focus is going to be on the learning objectives as per the application. You have to design and deploy these things in line with the robotic process automation and some of these processes that we will go through, right? Inshallah. Are you ready, Muhammad Irfan Alam?
Yes, sir.
Can you please share your screen before I start the timer?
Okay. Is it visible now, sir?
It's loading. Wait. It has loaded, yes. Okay. Bismillahir Rahmanir Rahim. You may start now.
Assalamu alaikum wa rahmatullah. My name is Muhammad Irfan. I study IO in Nafi International College. Today I will be presenting orchestrating synthetic biology automation platform with BioCAD, OpenTrans, and laboratory robotics for biotechnology research.
Go ahead, proceed, please.
Now let's get started. Let's get started with table of content. Introduction of synthetic biology. Project overview, automatic platform, why automation in synthetic biology. It is used to increase, improve reproducibility, speed, and accuracy. Key learning objectives, design, execution, and regulatory compliance. Overview of the DBT cycles. It integrates with design, build, test, and from biological design, architecture of synthetic biology automation platform, how to architect a cloud native platform for automation. Standard in synthetic biology ensures interoperability, reproducibility, and scalability. BioCAD tools for genetic circuit design, DNA assembly, and synthetic technology automates a synthesis and small assembly of genetic materials. Laboratory robotics, liquid handling for how to handle pipetting, sample preparation, high throughput screening, and analytical tools, how to analyze the biological impact, data management, laboratory information management, how to manage, store, and ensure the data, microfluid and real-time access, bioinformatic pipelines, data extraction and processing for data analysis and reproducibility, genomic transcriptomic and proteomic analysis for better performance. Artificial intelligence, synthetic biology helps to predict, help to predict,
Your,
helps to predict, automate,
your presentation is stuck here in the table of contents. Are you on the next slide? Where are you?
machine learning for predictive modeling, cloud labs, and remote experimentation.
Wait. I cannot see, I can only see table of contents. I cannot see other slides, right? You have to demonstrate to
Yes,
me the right slide. Can you please? मुझे जो नजर आ रहा है वो गलत नजर आ रहा है. I can only see table of contents, right? मुझे slide दिखाओ. Show me your slide, please. Now I can see introduction. Okay.
Yes, sir.
So if you are not on the same like slide, and if you're talking about something else and I'm seeing something else, I will not be able to give you score on that. So please do not make this mistake, right? This is the slide 3 that you have submitted. So make sure that you are on the right slide and tell them this is slide 3, introduction, then slide 4, project overview, then slide 5, so on and so forth, right? Please proceed.
Sir, synthetic
Continue.
biology and integrated biology. Synthetic biology and integrated biology with engineering principle and computer science to create or modify organisms for new functionality. Automation is transforming synthetic biology by improving experimental throughput, reproducibility, and scalability. The purpose to articulate a cloud native biology automation platform to integrate hard genetic constructs for AI modeling and ML processing, robotic and data system. The scope synthetic protein engineering, metabolic optimization, goals to reproducibility, scalability, and compliance. Why automation in synthetic biology? Automation in synthetic biology to improve accuracy, scalability, reproducibility, boost throughput, reducing cost and waste, enable scalable research, driven optimizations. Now, synthetic biology design automation helps to how to design genetic circuits, laboratory robotic integration, how robots help to generate genetic designs. Bioinformatic pipelines management help to how to analyze data for future purpose. Laboratory information management helps to how to manage data, store, and so on. Metabolic engineering optimization helps to optimize and workflows. Regulatory compliance and data management helps to ensure data security. Industrial application for drug discovery, biofuel production, precision medicine. There are multiple open source and collaborative tools such as BioCAD, OpenTrons, and so on. Architecture view, BioCAD for design layer, schedule the task, and their instrument used for how tasks to be performed in repetitive times. LIMS for data capture and LIMS used for data capture and management. Galaxy for data analysis and modeling. Cloud Airflow Mini for store data. Data is short or long depends on. First, erase square. It's a relational database. Store structured data. Compliance and collaboration. Electronic lab notebooks, audit trials before preparing a sample. Now move on. BioCAD and GenCAD used to genetic circuit and pathway design, DNA sequence and design optimization with the help of DNA box Taylor Jones. SBOL for standardized and interoperability. and Cobra OptiFlux for metabolic engineering pathway designs, and Deep Sun Python for machine learning and AI-driven tools for analysis and make accuracy. Plot-based and integrated platform such as benchling and DNA used for how cloud manage and make sure data is to be secure. Now let's move on. Platform OpenTrons is a platform where we automate liquid handling, integrate with PCR plate triggers, robotic scheduling for high throughputs. Now move on Galaxy. This platform for data analysis, sequence analysis, genomic protein, proteomics analysis, allogeneic workflows for uh, feature process. Now move on LIMS lab key server used for sample tracking how data is continued and processed. Experimental data management, data integration, chain of custody management. Now tools covered by optic flags. They are Python library used for constraint-based modeling, extent design, and screen pathway optimization for pathway design, pathway optimization, bioprocess optimizations to build our genetic circuits. Now, there are so many sectors we use biotech, pharma, academia, synthetic biology, startups. Tools like BioCat, OpenTrons, Galaxy Lab, Cobra by Sunbio. Infrastructure, Airflow, NIO, PostgreSQL, and standard FDA 21, CFR Part 11, GLP for regulatory compliance and security. Benefits of synthetic biology: it increase efficiency, productivity, reduce cost, improve safety, better compliance, enhance scalability. There are so many challenges facing while performing synthetic biology, such as biological complexity, issue with data standardization, high cost, facing safety and security issues during artificial process. Now move on. There are so many regulatory compliance used to secure, such as GLP compliance, ISO 13485, HIPAA for GLP, ISO 30, GLP workflow automate uh, pipelines, data privacy, system validation, audit trials, essential training, quality-based result, digital signature to secure our data. Now move on. User identity verification with the help of DA21 CFR Part 11. How to track action, who is accessed and who is not accessed by authentications. Version is 3. Which type of version you use while performing synthetic biology automation. Audit tracking retention, accessibility, how one who can access your platform. Audit trial, security, automatic time stamping. Now move on. Our data management system. So how to manage data, collect, store, and access. Essential for performance, scalability, and security. There are so many databases such as SQL for structure, query language, how to structure your data for future purpose. Now, conclusion to future trends, advancement of technology such as AI, cloud computing, etc., nanotechnology, digital twins, uh, that uses synthetic biology platform for uh, better product development, calculation, scalability, efficiency, innovation, and so on. Without a lot.
Okay.
Hello.
Sorry, can you hear me, Irfan?
Yes, sir.
Okay. So what is the DBTL cycle? I think I was on mute and I was asking you a question, and you should have said like I can't hear you. Okay. So what is the DBTL cycle? The in synthetic
Yeah,
biology,
it
and
is.
how, hold on, and how does automation enhances it? What are the benefits of that?
It is a closed loop process designed to result will test and learn for biological purpose.
I mean, what else is there? DBTL. What does DBTL stands for?
It's a design, build, test, and learn. How to
This
design
cycle,
a genetic.
this cycle is also used in pretty much, can be used for pretty much any application. Design, build, test, and learn, right? So,
Yes.
uh, explain to me the cycle. What is it?
Uh, this cycle is used to with the help of with the help of BioCAD for design genetic circuit, as well for standardized genetic representation and photon and galaxy for its analyze and modeling process for model the model the perform how to model the performance.
Irfan, when this topic, synthetic biology, is very huge, right? It is being used in many, many disciplines right now as we speak. Uh, you can apply in companies like bioinformatics automation engineer. They pay you a lot of money for this. Synthetic biology automation specialist, because you have to process, laboratory robotics engineer, biocad, workflow developer, right? So on and so forth. And this is highly paid. If you know your work, you can get a very high paying job. And we are now also offering specialization in synthetic biology as well as part of the AI of specialization, which we are about to launch next month. So I know this very well. When you have to provide me the answer needs to be like this. I ask you, what is the DBTL cycle? So you say, the DBTL test learn cycle is an iterative process used in synthetic biology for designing and improving biological systems. Automation enhances it by increasing throughput, ensuring reproducibility. You have to reproduce the same results, right? Minimizing manual errors and allowing real-time feedback from each iteration for faster optimization. Now, when you go in and work as a synthetic, engineer, automation engineer, your job is not to work on those things, but your job is to work on the system itself. Then the synthetic biologists or the biologists are using, right? So the answer needs to be better, Irfan. Okay?
Yes, sir.
It's not the best answer that you provided to me. Which layers are included in the synthetic biology automation platform? In the architecture that you presented. Explain to me all the layers, please.
So, firstly, we use BioCAD tool for design how to design genetic circuit, then move to SBOL for representing genetic way.
No, no, no. Hold on. Give me the layers name. There are five layers, 6 layers that you mentioned in your presentation. What are the layers? One is, I will start with one, design layer. The other one is
Test design build layers.
after design layer. What is it?
Build layers.
Sorry, your voice is breaking up. What is it?
After designing, we build it.
It's
Then
execution layer.
run
It's
and test.
execution layer. And after design, it's
Yes.
execution. After execution, what is it?
Then integration now.
Data management layer, integration. Okay. And after that, what is it?
data management and analysis now.
And analytics and modeling layer. And last one is
Starting with the model for better performance.
it's a cloud infrastructure layer. Okay. That is how you have to answer. Okay, so OpenTrons, OpenTrons. What role does OpenTrons play in the automation workflow?
OpenTrons is a open source platform um, for standardized representation of biological system now.
People, who works on these systems, they are normally engineers in bioinformatics, AI, and stuff like that. They are not like the biologists themselves, right? So OpenTrons is something that we have developed labs for, right? If I show you OpenTrons, right? OpenTrons is a system which essentially does what? So let's look into that as well, right? This is the OpenTron system. It's the benchtop liquid handling, right? So you will see lots of these systems. It's an open source liquid handler, improves your lab, uniquely flexible, and all of those things. Everything is now going towards robotics, right? So this is one of the things that are going towards robotics. So lab automation robots built for the future, right? So these guys are automation engineers. They're paid very high salary if you know how to get this thing working and fully optimized, right? That's what you have to learn. So, the future is now done and managed by robots and AI. That's why we are giving these exciting topics to our students to understand why they're important. तुम्हें इसलिए दे रहे हैं सब कुछ ताकि तुम्हें पता चले कि तुम्हें आगे जाके क्या करना है, ठीक है ना? जो इंजीनियर्स होते हैं जो इसके ऊपर काम करते हैं, the ones who are working on these areas, you know, let me tell you something quickly. Okay, in roles की salary क्या है? What are the salaries of these roles, right? Just to give you an idea. So, a bioinformatics engineer who works on these systems to maintain earns almost $120,000. And you can get a work permit in UK, Canada, USA, Germany, all these places. A lab automation engineer on average earns $110,000 to $130,000. Zero experience. If you know the software and if you can play with the software and get the work done. A robotics engineer who are working for automation and robotics and all of these things, right? And utilizing AI. They're earning $150,000 per annum. These salaries are huge, right? What kind of experience are they looking for? Zero experience. Because these are new technologies, they're looking for people who can work and move these technologies and connect them to the cloud. So your answers needs to be better, Irfan. Answers तुम्हारे बहुत अच्छे नहीं है मेरे भाई. They're not very good. Explain the importance of the synthetic biology automation. Why the SBOL, right? What does it stands for and why is it important?
SBOL stand for synthetic biology open language uh, for uh, for biological representation and creating a file with the help of this tool. And it's a programmable without any other. It is used to program biological systems.
Did you downloaded some of these softwares to test them out? How do they look like? How do they work? Most of them are Python based. Did you do all of those things?
Yeah.
Yes or
Yes,
no?
yes, but not is more times now.
Okay. You have to do some more work before we can move forward with these things, inshallah. Okay. Which standards are used to ensure regulatory compliance in synthetic biology automation? I mean, if you are going in as an engineer, a DevOps engineer, a cloud engineer, and you have to work with the regulatory compliance in the synthetic biology automation for the lab, what are the core standards you must follow to ensure that the systems are secure and compliant?
FDA 21 CFR Part 11 and HIPAA also.
So why do we use FDA 21, CFR Part 11? Is it for what?
It is used to secure food and drug development. We have to secure food and drug development, drug and development platform. How to
Yes, it is there to secure, but secure what?
it is used to secure their interoperability.
Not interoperability is interoperability means like different systems can communicate with each other. It is there to secure electronic records and audit trails. That's
Yes,
what it is. So that if the FDA, uh, if, the, if those agencies comes and they say, okay, we want to do an audit of your systems, then
electronic records are secure
yes,
now.
electronic records and audit trails. What does the GLP, good laboratory practice, does? Like, why is it there? To do what?
Good laboratory process are used to secure workflows now.
Workflows, laboratory procedures, integrity. Yes. Okay. Workflows. Good. What do we use HIPAA for?
HIPAA is used for in healthcare field. There are so many data regarding medicine patients. So how to secure their patient details, drug dose, discoveries, components.
Yes. I mean, they're there for data privacy and health information protection, right? And last one is ISO 13485. What do we use it for? This is all mentioned in your presentation.
It is used to cyber security risk, how to secure any ethical and any threats
No, no, no.
on the
It's not
system.
a ISO 13485 is not for security. It's for something else. If you give me the right answer, I give you the full marks, the last one. Otherwise, your marks will be deducted. Think, think.
It is used to secure the flow of data.
No,
Otherwise,
that's the GLP. This is for medical device quality system. Okay. Okay, let's jump in. So, as an engineer, when you go in and apply as a bioinformatics engineer working in the cloud and AI and all of these technologies, right? Um, describe how data flows through the architecture from genetic design to compliance logging. I mean, give me all the 5 layers and what happens between these five layers. If you want to look at your presentation, you can do so.
yeah, firstly it will be to digital design and use robotics for optimization and integration now.
So, okay, let's take a step back. You know, your topic is tough, so I'm going to give you some assistance. So, the first is design phase. What do we do in the design phase?
In design process, we design genetic circuit now.
The DNA circuits are created using
Yeah.
BioCAD. You know, some people, they call synthetic biology as playing with God, playing as God, right? That's not the case. Islamically, you can work with synthetic biology in many, many aspects, right? Uh, I will explain that later on. So design phase. The second is build phase. What do we have? What happens in the build phase?
In build phase means how to how to combine multiples of genetic circuit for better accuracy and performance.
Okay. Test phase. What happens in test phase?
Test phase in test phase we use automation platform to test uh, how much uh, uh, how much generative design will be performed for repetitive tasks, which is better for uh, future time now.
Okay. Hold on. Let me break this question into open my calculator, right? Okay. 0.5 plus 0.5. Okay. Learn phase. What happens in the learn phase?
As well as in genetic biology, collaborative platform learn the process and the created during design and build and
Okay. What happens in the compliance layer?
in compliance layer is to how much the platform to be secure.
No, it's like logs and audit trails maintained under FDA 21. That's what it is, right? Okay. All right. How does AI driven modeling like Deep Bio, or there are many other solutions as well that can potentially be used for this purpose? Some of the models are used in these phases are ones which are being used in UK as well in the US. So how does AI driven modeling enhances the predictive accuracy of synthetic circuits? I mean, how can it help us?
It helps to improve the model performance for better drug discovery, biofuel productions, and uh, precision medicine now.
So let me ask you, hold on, just making some notes in your file. Okay. What are the potential risks of using robotics in synthetic biology automation, and how can they be mitigated? I mean, this is some of the things that if you go in as an engineer, they might ask you this question. How would you answer that?
To ensure trust with stakeholders and
First tell me the risks. What are the risks of robotics? What are the risks?
it means to it's not be completely secure from threats.
I mean, okay, data integrity breaches. Okay, what else?
Data integrity breaches and threat threat on the system.
Cyber vulnerabilities. Okay, what else?
Next,
If your wheel is not calibrated, what will happen? Do you drive a car?
no, sir.
Okay, if do you drive a bike?
Yeah,
So if your wheel, if your wheel of the bike is not calibrated, what happens?
we should check out the what's issues has to be happening now.
So mechanical calibration errors, that's the third one, right? Because robotics in synthetic biology automation is doing what? It's picking up, testing, moving out the data, right? Moving out the vials and everything. This is what it's doing, right? The synthetic biologist doesn't know how to work in these robotics things. This is where you come in and they call Irfan, come And can you please check what is happening? The robots are doing something wrong. That's where you come in and say, okay, let me check. So there are risks. The first of the risks are cyber vulnerabilities, as you mentioned, data integrity breaches, mechanical calibrations, right? And I would say contamination that can happen. So these are the risks. Mitigation for contamination, regular sterilization, right? This can be done. For data integrity breaches, redundant sensors, data encryption. For mechanical calibration errors, robotic safety interlocks. And for cyber vulnerabilities, compliance with cybersecurity frameworks like NIST 882, ISO 27001, and stuff like that. That is how you have to answer. Irfan,
Yes, sir.
interview is not coming along very, very good. 3 questions remaining. Try your best. Okay? What is the role of airflow and MinIO in the data management architecture? Very easy. It's a slam dunk for you.
Airflow is managed simple data now, but employee used to um, store large data set for for better performance.
What is meant by ETL pipelines?
ETL electronic test ETL pipeline used to integrate.
In the context of airflow, what does it mean? In the context of airflow.
Airflow means to how the automation performance to be flow.
No. Okay. Let me show you to give you an example, right? So that you understand. The answers are not coming out very, very good. Okay, so I'm going to show you something. Airflow, Airbyte, these are all ETL pipelines applications, right? So what we do is like we develop, so let me show you. You see this, right? This is our Airbyte. Airflow and Airbyte both are ETL engines. What they do is like extract, transform, load. This is what happens in our internal systems, right? So imagine the data sources. These are all your synthetic biology, machinery, robotics, all of those things. The applications, they're collecting all the data through Airflow. And using Airflow, you create your own data lake. And through the data lake, then you create these dashboards. Do you understand?
Yes.
This is what you have to do. You need to work more in terms of getting to the point where you can explain to me better. Okay. How would you implement an audit trail for laboratory robotics data under FDA? I mean, how would you set that up?
Repeat again, sir.
How would you set up and implement an audit trail, for laboratory robotics data under FDA 21 CFR?
We set up after the design testing phase to secure with the help of FDA 21.
No. I'm asking like when you set up audit trail, right, for robotics data, what would you do? अगर तुम्हें robotics data का सारा audit trail, सारी audit logs चाहिए, तुम उसके लिए कैसे set करोगे? What are you going to do?
We are going to set uh,
Are you having difficulty in this exercise?
Yes.
मुश्किल है? So this is what you're going to do, right? When you set up the audit trail data, you set up in your topic, it is mentioned Postgres, right? So you mentioned PostgreSQL. And in the PostgreSQL, you set up the timestamps, the identity verification, the systems, logs, all of those things, right? Before coming to you, we have a big project known as Al-Basir, one of the names of Allah. In Al-Basir, we are collecting all the logs in our data lake, right? And through that, we are trying to make decisions of what we have to do. So similarly, तुम्हें इसी तरीके से काम करना है. That's what you have to do. Do you want to take another shot in terms of reappearing for the exam with a different topic? Normally, I don't allow that. मैं allow नहीं करता. You will be an exception. Irfan, I'm asking you a question.
Yeah, I
दोबारा
will try
एग्जाम
to do
देना
that.
दोबारा एग्जाम देना है?
Yeah, if you not got better score, then I go for again now.
दोबारा से बात इंग्लिश उर्दू में बात करो. स्टॉप द रिकॉर्डिंग
ये पे-
प्लीज. हाँ, ये
अगर
इसको
अगर
सबमिट
ये
नहीं
बेन
करना. ठीक है जी, बोलो क्या कह रहे हो?
अगर ये मतलब परफॉर्मेंस मेरा बेटर नहीं है तो मैं आगे के लिए फिर बेटर करने के लिए रेडी हूँ.
अभी भी एग्जाम देने के लिए रेडी हो?
ये सर ठीक है, अगर परफॉर्मेंस बेटर नहीं है तो फिर से ट्राई करेंगे.
मैं तुमसे कहूँगा, आई वुड रिकमेंड यू, आई एम नॉट गोइंग टू, you are not passing the exam right now, okay? uh, you can request a change of topic if you want, and we can give it to you, right? But right now you're not passing the exam. In the first 5 questions you only secured 5 marks out of 12.5. In the presentation the scores are not good, right? Normally when a person fails they have to pay the examination fee, okay?
Yes, sir.
I'm going to ask the Director of Academic Operations not to charge a fee from you and give you another different topic. Okay? Take your time. अपना time लो, 4, 5, 6 weeks लो. अच्छी तरह preparation करके आओ. You're a good student. You secured 70%. I was looking at your BS degree in mathematics and everything. ठीक है ना? मेहनती बच्चे हो, सब कुछ है. I don't know what is happening today, but it's not coming out very good. The topic is synthetic biology. The topic is related to all the internal systems that you have to design. This is not the synthetic biologist who does it, but bioinformatics engineer who does it, right? It's all open source systems, easier, compliance issues, all of those things. You are inventing answers. You're not fully prepared. Okay? I'm afraid you cannot pass this exam. We have to stop the exam now. There is no point. In the last 5 questions, you only secured 0.5. It is very difficult for you to pass the exam with this score, right? So you have to go back. Is Yusuf here? Huzaifa Yusuf कौन बैठा हुआ है यहाँ? Who's here? Someone here? I have to go back and ask them. So I think they made me the host, right? So you have to go back. I'm going to ask them to give you another topic. एक तुम्हें different topic दें. Go back, do your preparation, then come back inshallah. Okay? Irfan? The reason मैं ये कर रहा हूँ, मुसलमानों के लिए पहले ही इंडिया में बहुत मुश्किलात हैं. I'm not going to put pressure on you कि तुम जो है ना जाकर वो करो, ओके? और तुम जाके वो करो. My time is very precious, लेकिन there are exceptions that I always follow, ठीक है ना? And we have to do the best with the people. Looking at your academic record, it's a good record, mashallah. Okay? तुम बहुत अच्छा काम कर सकते हो. सही है? Inshallah. What do you want to do once you complete the AI ops? What do you want to do? क्या करना चाहते हो?
I want to sit in AI, artificial intelligence.
AI में काम करना है?
Yes,
There's a ton of work. बहुत काम है. ठीक है ना? यहाँ से जा सकते हो, सब कुछ कर सकते हो. इंडिया ने, आज सुबह मैं सुन रहा था, अमेरिका ने इंडिया के लिए मुश्किलात कर दी हैं. ठीक है ना? और उनको H1B वीज़ा में मुश्किलात हो रही हैं, राइट? फीस लगा दी. चाइना ने उनके लिए स्पेशल वीज़ा खोला है. So if you have good ideas, if you know your stuff, you can go and start working in China. They are opening their doors for Indian engineers. दुनिया की सारी बड़ी कंपनीज के सीईओ कौन है? माइक्रोसॉफ्ट कौन है? इंडियन. गूगल कौन है? इंडियन. ऑल दी ओपन एआई कंपनी के अंदर बड़े बड़े जो को फाउंडर्स हैं इंडियंस, राइट? क्लाउड के अंदर, आई मीन वेरेवर यू गो, वेरेवर यू लुक अपॉन, यू विल सी इंडियन इंजीनियर्स. दे आर मुस्लिम्स, नॉन मुस्लिम्स, बोथ, राइट? बिकॉज दे आर हार्ड वर्किंग. ठीक है? सो आई एम गिविंग यू अ फुल अपॉर्चुनिटी. मैं इसको मार्क नहीं करूंगा. दिस विल नॉट गो ऑन योर रिकॉर्ड. जाओ, मोहम्मद इरफान आलम, प्रैक्टिस करो, फिर वापस आना. ओके? I will ask them to give you another topic. Okay? I don't normally, मैं करता नहीं हूँ. I don't normally do that. Right? But there are exceptions. ठीक है? Your academic record is very good. तुम्हारा बहुत अच्छा record है. Okay? So, you have good potential. Today is not your day. It happens. Okay? Inshallah. Jazakallah khair. Parents zinda hai?
yes, sir.
Ask them to make special dua for me. Inshallah. Okay?
Yeah,
मेहनत
you
करो.
also need to us.
कोई मसला नहीं. अल्लाह ताला आसानी करेगा. Work hard. When is your AI ops expiring? कब expire हो रहा है तुम्हारा?
I soon want to comment now.
नहीं, अप्रैल में लिया था ना. अप्रैल टू अप्रैल. मई, जून, जुलाई, अगस्त, सितंबर, अक्टूबर. अक्टूबर में खत्म हो रहा है तुम्हारा. 6 months पूरे हो जाएंगे और एक्स्ट्रा, राइट?
Yes,
So you have to acquire additional time to complete the labs. The labs are very important for you. अगर तुमने labs complete कर ली, if you complete the labs, you will be unstoppable. Inshallah. You can go to UK, you can go to Canada, you can go to Australia, Germany, wherever you want to go, inshallah. बाहर पढ़ने के लिए भी जाना चाहते हो?
yes, sir. So how much time to take to complete all the labs?
At least 6 to 9 months. 9 months. 6 months are the first 22 domains, and the last domains, 23rd, is different. It will take you time.
But my enrollment is to be going expired soon now.
If that
What
expires,
is
then you can't do the labs. You have passed the diploma, but you can't do the labs. I mean, labs are something separate that we just recently added. We can't go back retroactively and give it to the old students. We can't do that, right? So for that, you have to maintain an active वो. ये तुम्हारी अपनी मर्जी है. If you want to do it, I would recommend you to do the labs. The labs will give you the confidence you need. Installation of these things. देखो, दुनिया में कहीं कोई चीज कामयाब होनी है ना, द फ्रेमवर्क, द स्टैंडर्ड्स, ठीक है ना, द रेगुलेशंस, द ओपन सोर्स सिस्टम्स, ऑल ऑफ दीज, इफ यू हार्नेस ऑल ऑफ देम and the job description, you can secure that job. That's exactly what we do. So you will be given extra time to pass your exam. I'm going to ask that from them. But when your portal expiration, when your portal expires, then I can't do anything for that. You have to renew your license, your access to do the labs. Whether you do it like, and the Razak program is released month by month, right? अभी तुम्हें नजर आ रहा है ना, you can see the Razak, right?
Yes, sir.
You should do the labs. If you want to do it faster, it's up to you, but it will be released month to month. We can't give it in one day. हम एक साथ नहीं दे सकते. That's not possible. We don't do that, right? Anyway, Jazakallah khair for your time. May Allah make it easy for you. I'm going to stop the marking here. This exam didn't took place. There's no need to go. This is not going to go on your record. Your record pe nahi jayega. Okay? This is not going on your record. So don't worry about it, inshallah. Go back, prepare yourself. Inshallah, okay? Chalo. Jazakallah khair. Subhanakallahumma bihamdika. Ashhadu an la ilaha illa anta astaghfiruka